Counting DNA damage foci gives us a quantitative measure of DNA damage, allowing us to compare how cells respond to different treatments or conditions. Here I have a few images from a user where cells were exposed to 2 concentrations of a DNA damaging agent and stained for nuclei and foci, and I've built a workflow to quantify the number of foci for each nucleus. First, I've created a segmentation mask for the nuclei using segment cell nuclei, and I filled the small holes within the masks using fill holes. Then I've segmented the foci. I applied an FFT high-pass filter to make them stand out a bit more, and I chose overlapping background segmentation as the actual segmentation strategy. You can see that sometimes when 2 or more foci are very close, they get assigned the same label, so I used the pick seeder followed by flood fill to separate them. Finally, I used associate objects and join sub-objects to count the number of foci for each nucleus. This is where you find the results. Here I've plotted the area versus the perimeter of the nuclei, so we can use this plot to gate out any unwanted detections. For example, if you draw a gate around this dot, you can see that it is an aggregate, so you can create a gate to exclude it from the analysis. In this dot plot, you see the number of foci per nucleus, while this bar chart shows the mean number of foci for each cell group, and you see that the number of foci is proportional to the concentration of the DNA damaging agent.